Hey guys, so this video is on monoclonal antibodies. This is our last topic in the immune system. So the learning intentions of this video is to know the purpose of monoclonal antibodies, to understand how they're made, and to understand how we might be able to use them for treating cancer. So monoclonal antibodies, well, what are they? Well, they're defined as an antibody produced by a single clone of cells or a cell line, and they consist of identical antibody molecules. That's where that term mono meaning one and clonal meaning clone comes from. So they're a form of artificial passive immunity. So that means that if we take these antibodies, we have no long-term memory. It's just for that short-term ability to attack off certain antigens. And so that fits into the category of artificial passive. I'm going to go through how they're actually made and what we use them for. So first thing that happens is we actually inject an animal, and most often it's a mouse, with an antigen. So I'm going to use chickenpox as an example in this video. So we inject it with an antigen that has, say, for the chickenpox, and that causes the mouse to start creating antibodies against it. And we know that the cells that make antibodies are B plasma cells. So the second step is to remove those B plasma cells from the spleen of the animal. And so we've taken out the B plasma cells and we combine them with tumor cells. And the reason why we combine them with tumor cells is that tumor cells can rapidly grow. They've got an ability to replicate really, really fast, whereas plasma cells by themselves don't have that ability. And so when they fuse together and they successfully fuse, they're known as hybridomas or hybrids. And so they've got sort of the best of both worlds. They've got the ability to rapidly divide from the tumor cell, and they've got the ability to make antibodies from the plasma cells. And so what we do is we grab those hybridomas and we grow them in a medium, and then we pick the exact ones that we want that have made the exact same antibody, or the antibody that will match back to the original antigen, in this case, chickenpox. And so we take those hybridomas and we grow them in a culture, and then we harvest those antibodies for use. So in other words, we're basically doing what the immune system does in a petri dish and taking the final product, which is the antibodies, and we're going to use them for a variety of different reasons, and we'll go through what they are. So just to recap, we inject the mouse or the animal with the pathogen or antigen. We take out its plasma cells that are starting to make those antibodies, and we combine them with cancer cells to form hybridomas. And then we pick and choose the hybridomas that we want, and we end up harvesting and growing a very large amount of antibodies in a very short amount of time. So again, here's just another simple diagram. So here's this looks like a hamster or a guinea pig has been injected. We're taking out the cells from the spleen, those B cells, and we combine them with a tumor. We go through the fusion and we pick the exact ones we want, and eventually we take the antibodies that they produce. So it's like a little farm, I guess, to get antibodies in a very short amount of time that are highly specific against the original antigen that we started with. So the benefits, well, it means that we can make a large amount of antibodies in a very small amount of time. And those antibodies are highly specific, and it can be done in the lab. So we don't require a human to actually go through the immune system process, and there's nothing involved. It's just the ability to do this in a much smaller scale. And we then use those antibodies to treat cancers, to diagnose diseases. There's a variety of uses for them. The main one we'll talk about, though, is the treatment of cancer. So you can see here, we've got a cancerous cell, and it's showing these antigens saying that it's cancerous. So the monoclonal antibodies actually are specific to those antigens, and they attach to them, and that will tag to a macrophage or a T cytotoxic cell to come and destroy this cell as quick as possible. So we're using it to almost enhance the immune system and identify which cells have cancer or are cancerous. So Herceptin is an example. This is the name of one that's actually produced in the real world. And it's just an example of a monoclonal antibody. So it would have gone through the same process where there was a mouse or whatever animal they've used, and they've injected it with a particular antigen. It's made antibodies, and we then extracted the antibodies. So this particular one, you can see here, this orange monoclonal antibody just binds to certain proteins on the outside of a cancerous cell. And so when they're attached, we know that's really, really effective for macrophages and T cells. In this instance, it's T cytotoxic cells, to come along and cause that cell to be destroyed. And so that's going to hopefully be able to destroy that cancerous cell and limit the ability for the cancer to spread. So this is an example of how we would use it as a cancer treatment. So obviously there are many pros, but there are some ethical issues with using monoclonal antibodies. The first of all, obviously, is that we are using animals in the process. So most monoclonal antibodies are developed by using animals as the original host. So whether that is a guinea pig, whether it's a primate, whether it is a mouse or a rat, we are using animals. And that poses the question of, is it right or wrong to be doing that? And what sort of welfare is that for the animal? The second concern is genetic engineering. We often have to put human genes into the animal that we're testing on. And so that's the idea of playing God and being able to move our genes into other animals, and what dangers does that pose? And again, is it ethically right to be able to do that? And what things we need to put in place to make sure that it is safe as well. And lastly is human safety. There have been some cases where monoclonal antibodies have been injected into a patient to try, and it's actually resulted in their death. So again, is it ethically right to be manipulating all these different genes and animals just to have it in the end actually be harmful to humans as well? So it's a good thing to think about ethical concerns. We're talking about some of the topics we're going to come up with our next unit as well. So in review, basically the monoclonal antibodies are produced when we clone a specific cell known as a hybridoma. And we know that that hybridoma is just a combination of B plasma cell and tumor cell. And we've got that together, and they end up making really specific antibodies that we harvest. And we then use that for things like disease diagnosis and cancer treatments. So that's just a basic wrap up of what monoclonal antibodies are. There is going to be, I guess, a couple of more in-depth things that we'll cover in class as well. Thanks.